Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Ready to go? Very good. Good morning. My name is Lee Darks. I'm the Director for Education and Scholarly Communication in Microsoft External Research. And I'd like to welcome you to this first session of the morning. And I think I'm going to keep introductions quite brief. This is Dr. Jeremy Frey, Professor Frey. And we're very interested to hear about what you and your colleagues at Southampton have been up to. So I'll hand it over to Dr. Frey.
Thank you. Well, since a significant number of the audience are from Southampton or had something to do with this work, it's going to be very interesting, but we'll see what we can. But Carol hasn't seen it. You could have waited till the end of the lecture to have spoken to the speaker this morning. It's these northerners. Okay, so I'm going to talk about some work that we've been doing with our laboratory blogging system and linking it up to the MyExperiment system. And it's work that's been done by Andrew Milstead in the audience, who's behind what was called the Lab Blog system, and now we've formally calling it Lab Trove. And a number of people on the MyExperiment side, and particularly Danius from Southampton, who's been instrumental in coordinating stuff between Lab Trove and MyExperiment. And I will try and give you a bit of background as to why we're doing all of this, as well as actually some of the implementation. And I've just been asked, even by some of my colleagues from Southampton, is that really a picture of Southampton? And it is. It is actually a picture of Southampton, but I thought a small change here might be necessary to put us in the right location. Unfortunately, Southampton doesn't quite look as good as San Francisco. And this morning I was up and I actually hopefully will be able to change that for a picture which has the sunlight reflecting off some of the San Francisco towers, but we didn't quite. So I'll just talk a little bit about laboratory and notebooks and where we are with digital notebooks, electronic laboratory notebooks, and so on. And in particular, the idea of laboratory blogs and the blog books, perhaps also setting that within the context of the sort of thing that Adam Bly was talking about yesterday. And then to the meat of it, to show you something about the link up with MyExperiment. And maybe I'll change some of the slides quickly with Carol can make comments on the MyExperiment ones, so it is a very interesting year. This year, 2010, is the 350th anniversary of the Royal Society, and there's been a lot going on in that in the UK. And I've put that up particularly because in 350 years ago, two interesting things happened. The Royal Society was founded for a place for discussion, and at least in principle, open discussion, at least if you were a member of the right social caste in the UK. And actually, Robert Hooke was the person who wrote most of the stuff down in the early days. Also, Samuel Pepys wrote a diary starting then. That's an example of rather more closed, as I don't know how many of you realize it was actually written in code, in cipher. And I actually wanted to get a decoded part that might have illustrated why he wrote it in code. The documentary version of his diaries that was on the radio did seem to skip over the bits that I understood was the reason why he kept it quiet. But he was an interesting character. And these days would probably have been done for fraudulently acquiring a lot of government money through interesting naval contracts. And the current Royal Society building in Carlton House Crescent, if you're an exalted member of the establishment and do really well in science, you get to go and pay money to be a member. You should understand that fellows of the Royal Society do actually have to pay to be members as once you've been elected. 100 years ago was the 100th anniversary of Dorothy Hodgkin's birth, of protein crystallography. 70 years for penicillin, 50 years of the laser. Don't know how many. That to me is quite surprising. It's 50 years. 20 years ago for Hubble Space Telegraph, and 10 years for the human genome. And I think that also just the implication there in the volumes of data is another way of looking at that, the exponential or super exponential growth in data and types of data. All of it, though, towards the end, ending up in digital forms as the only way you can really manipulate that. But increasingly, that is the way it is. It's not that it's a picture that is represented in digital form. It is digital. That was what was recorded. There wasn't an analog part to this at all. Now, this has led to some interesting problems. And the issue about what scientists are supposed to do, what they were told they should do, what they tell other people they did, and what actually happens in the lab is getting to be quite controversial. Because we're all human, we do the best we can, but if I come and ask you at your home to find a telephone bill from 20 years ago, some people still have them, and if you can find them, it's quite amazing. But we expect that level of ability to trace in principle in science. In practice, most laboratories couldn't do that, and even if they can come up with the data, you can't read it. Provenance information has been lost. And in the UK, we've recently gone through this escapade of ClimateGate, hacked emails, misinterpretation of the emails. But in the end, I think the most interesting investigation of that was the Royal Statistical Society's investigation about what had actually been going on with the data. Was there indeed any evidence of any manipulation, unwarranted manipulation of the data? And the answer is no, but equally well, there were several comments that they were not using the best statistical knowledge that is available or was available at the time because no statisticians were actually involved in it. So there is a big break between, which frequently happens, that statisticians get involved after the fact, if at all. And most of the scientists think that they know how to model the data and don't consult. And they do a pretty good job, actually, but a better job could be done. We've had some interesting issues that's enabled StructureGate of protein crystallography data that's fraudulent, small molecule crystallography data that's also not what it ought to be. Indeed, in that case, the fraud was perpetrated or perpetratable because raw data was provided, or more raw data was provided, so it was easier to fake a new experiment. But it was also easier to detect it ultimately as well. The take-home message, I think, from the BBC website, of course, the highest accolade you can get in the UK is to get on the BBC News, Technology or Science website. That reaches more people, as we've realized, than anything else around in the UK. But their comment here, I think, was very reasonable. What do we all get told at school, and I think they still get told this, show your workings. We certainly tell our students this. The right answer is important, but how you got there is just as important. But how many times is that honored in the breach in most scientific publications? Now, one way we went about dealing with this was some work we actually did with Monica, MC, to look at a very strict version of a laboratory notebook for organic chemists. And that work is still, I should say, going on, but slowly, because a lot of the chemists couldn't really get on with that. What we could get on with a bit more easily and a bit more widely is a blog mentality. Now, when we started this, none of the science blogging stuff that we heard about yesterday was really active. None of the blog systems that were around would really support the necessary conversation that you wanted, even with yourself. Now it's a bit different, but at the time we started with some blogging, and basically Andrew Milstead and Cameron Nayland was really one of the very keen colleagues who wanted to deal with this. Basically had to say, well, we'll have to write something ourselves to be getting on with. We're hoping that's going to change in the future, and we might get some more supportable code with translating these things into some of the Microsoft products, but we'll have to see how flexible they are. We'll push the limits everywhere. But there are these blogs now. These tell you about science and comment on science, but they don't actually help you do the science in the lab. They're not designed for that. They're part of a different part of the scientific life cycle. Interestingly, one can ask how some of these things are supported. And one of the comments from last night, really it's summarized in the old thing. He who pays the piper calls the tune. And if it's the state, well, if it's the people via the state, then maybe they think they have a right to get the information back. There are some states that would not consider that was in any way a right. The state owns the rights to do what it wants with it. So does the public. So do the private individuals who might fund things, perhaps. So when it comes to supporting these blogs, most of them are supported by advertising, which we could have been difficult last night and asked him about Pepsigate, which was Adam's attempt to secure a stream of funding through the future, which got him into a lot of trouble. Now, this being a Wikipedia entry, I have no idea whether I can trust. Actually, no, this one is, I think, one I got from Wikipedia in the newspaper. But I have no idea whether it's really true that all his bloggers left or anything like that. But it's symptomatic of a major problem, which in a sense he was alluding to into paying for the dissemination of information. He has the same problem. He may be rude to the other publishers about they better change their models. It's not clear that his model is supportable either, which is a problem. But of course, if we were all in a more equitable society, we would just be paid to do our job, and somebody would be paid to disseminate it. But that has shown to be perhaps less than successful in other ways. But you never know. Now, the issue of fraudulent material, factual error found on the internet. The Onion is my favorite thing. It's finding a correct thing on the internet and how do you know is more of the issue. But in the journals, okay, so there's a whole system of reviewing that's supposed to take place. And actually, the journals are very strict about the information that is supposed to be provided. And if it is all provided, it does make it easier. Some of the journals in, actually I'll get this one, in organic chemistry are very strict. But actually, again, is all the information always provided? Is it provided in an intelligible form? There are one or two journals that require the reviewers to actually undertake the procedures and make the compound and check that it works, and often it doesn't. There's only one, as far as I can tell, there is only one domain in which this is utterly routine, and that is in mathematics. A proof is published in a mathematics journal only after the reviewers have actually reproduced that proof and checked it. And frequently, the dialogue between the reviewers and the authors is extensive because the reviewers will find a mistake, but it's usually not fatal. It can be got round. But that's just normal, but it takes 2 or 3 years to publish a mathematics paper. The rest, the peer review is it reasonable? Does it seem sensible? If it's surprising, what have they given us supporting evidence? That's not the same as repeating it, but it's the best that we can do. But if we escalate the speed of science, then maybe we do have to watch for that being a problem. But when we come to actually providing the evidence for this, a lot of it comes from the notes that we are taking in the laboratory, the notes of our observations as we're taking them. And Faraday's notebooks are an inspiration. They're an inspiration to me because he couldn't spell either. But what he did do was to provide a really good description of why he was doing something. What was going on? How did he get to this idea? Then the experiment. Nice sketch, an experiment, some results, go on from that. It really tells the story, the real story. And the story actually can be very helpful, especially to people as you start in a new area, about how to think about problems and so on. And Peter Day has produced these, and they're available in a book, and some are online. But there's a lot of interesting advice out there about how to keep notebooks from our colleagues in the University of Belfast, which tell you research books should represent a diary. Well, I agree it's a diary, but not have the data in it. Well, actually, I think that's wrong. Where is the data going to be if it's not associated somehow with your diary? It's the diary is the metadata describing the data, because inevitably you will forget to put some important metadata down. Let's be even nicer, you won't know that that metadata was important. You'll have no idea. But some of it can at least be reconstructed or thought about from the diary entry and what you were doing why you were doing things. These things are critical. They must appear together, logically together, but they don't necessarily have to be physically stored together. So we've produced, Andrew has gone through a number of generations of the laboratory lab log system. And these are just some of the style sheets, stylings of the different, some of the different blogs that are in operation, some in Southampton, some in Australia, and some are propagating further out into this country. And we even offer some sort of hosted service on the Our Experiment site, because we're very our sort of people rather than my people. And that's just an experiment to see whether it was possible to run from a central server based in the National Grid Service. And it is actually the service one of the University of Sydney runs is hosted in the UK. And it does just about work until somebody cuts one of the cables going through the Red Sea and things slow down, and that does cause a bit of a problem. There are a couple more there we have. And I did have to thank Cameron Nayland for a huge amount of inspiration and pushing us to produce more and more things. And also for the fact that he runs a fully open laboratory in a daring way that most of us wouldn't wish to do, but he doesn't mind. And so whenever we need examples, there is always an example available online and readable, and we can find out what Cameron's up to, which is useful sometimes when you want to get him to do something as well. So let me just give you an example of the sort of stuff that Cameron does. One of his students who was started out on this trek was doing sort of microarray type things, a lot of DNA PCR reactions and relatively complicated sequence of things, often done multiple experiments at the same time, and they would use different bits of DNA, the primers, mix it with some other stuff, replicate a sequence, and represent it in the end as a table where all of these entries link back to the blog post that describes that particular primer, and the blog post that gives us the sequence at the end, all completely linked up. These tables are then very powerful, but they're quite tedious to construct and get all the links in. You've got to have created all the posts and then link them all up. And so the idea of having something like this where you do get all the information in the table and you can see the product, the conditions, the reagents, everything is linked two-way, so when you discover that a whole series of reactions didn't work, you can trace it back to the fact that one of the primers was not what it said on the label, and then immediately you know all the experiments that should be looked at again. So everything is linked up, and to some extent is machine interpretable with a little bit of work. So the idea was, can we make this easier? Building up these tables is, as I say, complicated. So if you can make it a little bit easier by providing a template so that you can generate the template once and then easily reuse it. So every different type of experiment, these are often repeated many times, you can just simply reuse the template. And here's an example of setting up and editing and linking up some of these tables to make the template pictures in them as well. So this is one of the sorts of things that the template actually looks like. A set of codes that are relatively easy to write. That's the visualization of the template. Tells you whether it's going to be a table cell or a box or a drop-down menu. By using some of the metadata keywords, key value pairs that we can put in, and some of those can be also pre-prepared in the template. If you say a column is going to have DNA in it, it will give you a drop-down list of all of the posts in this blog that have DNA as one of their keywords. So it becomes a filtering mechanism as well to make it much easier to link up to the right systems. Now, if you've got these, it provides in principle a relatively simple approach to creating and using some semantics. This isn't a semantic system, but it does have a lot of metadata added to it. So you want one of these things, generate it. It is this metadata issue. And so if somebody spends some time working out this particular type of experiment, maybe a characterization experiment or one of these DNA experiments, this is the information you need to record. Get that sorted out, get out the table working right, get it formatted really nicely, all the rest of it. That's quite a lot of work. You'd like to be able to share that. Well, sitting inside any of the blogs, no problem. It's sitting in a section called templates. You can just go to that blog, copy it in, no problem. But the idea is you'd really like to share that a little bit more widely. And, I should say that having done that sort of thing and got it represented in XML in some form, lots of things then become possible. For example, using the MIT Simile software, you can export this XML into a timeline or an exhibit view, or even Steve Wilson has managed to put the timeline inside an exhibit for looking at making it possible for me to find out which experiments have been going on recently, and going particularly, he hates me when I trawl back through the past to find out some data. Why is this data different? And if you watch Cameron's Twitter feed, you can discover that he can spend several nights converting links from one form to another, sending it off to somebody else who produces visualizations, and then they talk about it for a while. So we've got these templates, and that's very nice with the templates, but we need to share them. So it sort of perhaps is a technological breakthrough to fix a previous problem or a previous breakthrough, because in the past you just drew a table on a piece of paper and somebody could copy that. Now we've got a more useful thing, but it's more difficult to exchange. But fortunately, there are other people thinking about how to exchange procedures as well as data. And Open Wetware and Useful Chem people, Jean-Claude Bradley, had talked about exchanging procedural information. But procedures are, in a sense, a generalization of a workflow, or at least it's the original meaning of a workflow from the industrial environment, because workflows involve people doing things as well as computers doing things. And so we have, of course, a nice system around Southampton and Manchester. And before my colleague Dave Giroir defected, it was very easy to go and get information on what was going on. And if he would at least have defected to an international, gone to another country, I could have ticked all those boxes on our EPSRC forms that said with an international author. But I couldn't. He's just gone down the road, or straight north, actually. So my experiment was designed as a way of sharing workflows. Originally just the Taverna workflows, but then has gone on to share almost anything that can be loosely packaged up with a workflow and have a picture. So Facebook for Scientists, I'm waiting for the first general release of the film describing a development of my experiment as opposed to Facebook. I think it could be very interesting discussions there. We haven't yet had a discussion on who should be the leading actor and actress, but I think we can work on that. Scary enough, yes. Fortunately, we now have Mark Balkan, who was one of the originators of the software for my experiment, working for us. So if we take it over, we haven't told Dave about this yet, but the idea is to share to workflows. But of course, you have to have lots of things that are implicit in that. Putting them there, knowing they'll be safe there, characterizing them, indexing them, searching them, so on. Otherwise, this is just pointless putting them there. People won't accidentally find them. So all sorts of things are needed, but it does mean that those workflows can be reused, and there are some very nice examples that I'm sure many of you have heard Carol talk about of Taverna workflows for analyzing gene pathways and drugs and so on that turn out to can be reused in other contexts and discover similarities between disease pathways that were not previously known. So it is sharing pieces of the process. So we've actually have some chemistry infiltrated into my experiment before it, and this was in a traditional route, just describing it. It's not a computer workflow, but subsequently Peter Lee's done some work with John Paul Bradley and has got some Taverna workflows that are chemistry ones. And once you get things into my experiment and so on, there are all sorts of other widgets that can be built to do things, so we can take a lot of advantages of the stuff that's already there. Because, as we know, plagiarism is certainly the name of the game here. Don't do it if somebody else has already done it. So what do? We needed to work out what packet of information needed to be exchanged, and fortunately, we have in the lab blog system, an XML export. So the easiest thing to do to get around any of the issues of identity and breaking security is to download an XML export of any post, in fact, but the sensible ones are to do it with the templates. Otherwise, you're exporting information as well. Now, you may wish to share the information, or you may not. But the template is the main thing. So you take out the XML export, and that contains the information which will describe this template. And I should say this is an example chosen here is from a proton NMR spectrum that you're recording the data and characterizing. So this is a pure chemistry one, and it has a suitable table here. It has links to previous posts, but it also has quite a lot of metadata in terms of key value pairs that would be the sort of thing needed to characterize. A chemist would want to know about that NMR experiment. So they're there. People know that they should fill them in. There's some of the XML, and just to highlight some bits, that's actually the code for writing the table in the blog. That's the bit that gets does the action, does the work inside the blog. There are the bits that describe the various metadata key value pairs that you're going to need. There is the HTML that renders the picture, and there's the picture. The most difficult thing is the picture. Deciding on what picture should be the key thing for my experiment, so that you actually have some view of what this object is when you're scanning the pictures, because those are the things that people look at. That turned out to be the most difficult thing to decide what to do and then how to get it. But the whole package is there. It's then, you log into my experiment, trying to remember your OpenID address and get into it. You can then go to upload the workflow, and in there, if you've got keen eyes, you'll see things that will actually say blog 2 template at some point. That's now going to be called Lab Trove, and it should have changed. You upload the template. You can search for them. There are a number in there, and when you find them, you can download it. You can actually, well, first of all, look at it, see who put it in, and see what descriptions were attached to it. All the sorts of things for any workflow. Now, quite a lot of those, the basic things, are extracted directly from the XML package that you loaded up. And as we improve the metadata of that type of metadata in the lab blog, we can update the filtering system in my experiment to take more of that out. But there's plenty of opportunities for putting other more detailed descriptions in that will help people find and use the template. Once you found it, bring it down and put it into your own blog and use it. And you see the code then draws the table, and you get your drop-down menus and so on. Now, we've been working on these things for some time, and just to give you an overall picture by way of introducing some of the talks that are going to come up from a group later on. So say we started working on with the CombiChem project in what were the Smart T projects, and looking at applying quite strict semantics to a specific area of organic chemistry, and that continues on. And that fed in terms of our understanding of RDF and the project that comes from Microsoft OREChem, to understanding critical idea really turns out to be the plan. And that's where we're going to take things forward in the future, that if you've got a plan, if you do things properly with a plan first, all sorts of things then can happen automatically afterwards. And that's coming through, and Mark Borken will talk later on this morning on the OREChem experimental ontology. And linked up, that actually follows through to be used and consumed by a new semantic blog system called Blog 3 that Mark is generating. Meanwhile, Lab Blog system has undergone some extensive user trials. We've been using, some of the groups using it, we've got more or less 3 years worth of data. It's getting quite impressive, the amount that's in there, and how useful that still is. In a weekly meeting I run on one particular project, the ability to go back and find stuff. And that has modified some of the aspects here, and we got some money from the University of Southampton to start to bring this together as a proper open source project for download and to go towards a more commercialization. And that is happening as we speak. And indeed, there is a LabTrove website and a SourceForge site. So if you go there, you can actually download the LabTrove system, which will have this XML export to allow you to upload to MyExperiment. Now, all of this is really very useful, and it allows us to take all the places where people have discussions and bring to the discussions all the critical data and the blog. And there, you don't have to write a special presentation for your group meeting. It's all there. But fortunately for us who drove past the Claremont, so I was a postdoc here in Berkeley between 82 and 84, and I drove past the Claremont on a number of occasions and looked at it and thought, that must be a very nice place. So fortunately, it is still necessary to have real meetings so we can come to places like this. And I thank Microsoft for hosting these. And I just finished with a thought that has come up in a project that I'm involved in Southampton, which is actually a high-resolution computer tomography, X-ray tomography set of experiments from our colleagues Ian Sinclair, when we were talking about computation-intensive imaging. And there is an issue for the scientists about intensive. Intensive properties are ones that do not depend on the size of the system. Extensive properties are the ones that actually technically scale proportionally to the system. So it certainly should be data extensive science. Whether there's data hyper-extensive or something, I'm not sure. But I think whether I can trademark in enough time for Tony's second edition should be data extensive science as the fifth paradigm or the fourth paradigm. We'll have to see. To just acknowledge the people who did the work, particularly Andrew and Danius, this project, and Cameron and Dave, for the linking up between the LabLog and MyExperiment, funding from JISC, EPSLC, and Southampton. And of course, it couldn't have happened if MyExperiment wasn't there, and there are some of the acknowledgements for MyExperiment. Thank you very much.
Thank you. So what I'd like to do is ask maybe a question or two for Jeremy, and then we can maybe transition the computer. So I'll invite Michael up. So are there any questions for Jeremy?
I have a question.
Oh, okay.
Reluctance of scientists to use the shared lab book, to share it with you. Just have you convinced the chemists to use it and to share the data? To share the lab books, not the data. That's a different.
Yeah. So we have a couple of chemistry groups using it. So physicists, some engineers, and some bioorganic chemists. So it ranges. You can set the security to where you want it. So we have Cameron Nayland running a bio lab in by the ISIS neutron facility, and it's a completely open notebook science runs for some of his things. We have a physics project where it's used at a slightly higher level, not their individual lab books collectively, and that's shared, but just amongst the project. And then we have other people who use it as an individual notebook where only they and their supervisor would have access to it. And again, it all depends on when you want to make things free. What it does mean is that when you publish something and you want to make it available, you can, but prior to that, you can keep it closed. I believe that you have a my personal belief is that when you publish something, you have a responsibility to make the data behind that available. But I'm not a zealot that says you have to make it available the hour that you made it, when you actually collect the data. And so it was a requirement of the system. You could tune that availability. So run it completely on a separately contained server, no access to anybody else if it was important to do that, or run it all the way through to completely open. And as we develop the system further, it's even going to be possible to have individual posts can be closed or open. So the advantage there is, suppose you do want to run a blog that's publicly available showing what's going on in your lab. You probably don't even then want to have everything that goes on in there, but you might mark some posts as ones that could be made available to outside, and you will be able to do that. So that fine control is necessary. Comes with it a bit of overhead, you have to decide to do it, but on the other hand, if somebody phones you up and you want to write a press release, that's a lot of work, whereas if your institution can see a few selected ones that's going on and they write the press release from that, you can benefit. So depends on circumstance. But some chemists are beginning to use it, yes. We're not all like the ACS.
Another question here.
Hi, Jeremy, how is this relating to the vision that you've had for a while about chemists, when they're publishing work, this is the hot stuff they want to publish anyways, that table they're producing? How is this going in terms of connecting with the whole nature of publication and what we were talking about this morning with respect to credit for this kind of work? This is substantial to do. Tables are still a pain in the neck, can take time to produce. So what are you seeing as the connection now?
So whatever you do, to produce that finished publication or presentation that looks good and communicates the information is hard. I do agree with Adam Bly that the designers have something to add here. But what we can do here is at least you can say, right, the data is there. That information is there, and we can link all the individual elements. So Steve and my group has run, say, 600 experiments of a certain type, and each one is in a database, and the table of data that we feed into the analysis program has a column in it with the URI of the data. So everything transported all the way up. So finally, when you'll put a spreadsheet, even if it's a spreadsheet attached to your document in the paper, and the links will be there. And those then, we can also run the same sort of citation tracking that you do between papers, can be done so that if you've got a URI of your data item, and they get back to that in a paper, you can then find all the blog posts that he made about that as well. And this is something that you would then open up after publication. So it doesn't make it any easier, really, in some senses, to write the paper. That's still, the intellectual effort is there, the crafting of it. But you don't then have to say, right, now let's move that data the fundamental raw data we want to link to somewhere. It will be on a database that at least will last as long as the institution will keep that software spinning. And so those links will be there.
In your community, what's the energy been to have what you're doing become the gold standard of practice?
All right, yeah. Okay, so I think the answer there is it's beginning to be seen to be useful, but it's just beginning. So some people do it, and they do see their data being used. And the biggest impact, the direct immediate use, is when you're teaching and you actually want to find some real raw data that people have processed, because this is really quite hard to do. But that, of course, means that that paper gets cited into a very much wider audience and best practice is seen. It's slow, but in the chemistry, we don't do preprints. We're starting, well, you heard what the ACS is like. But the Royal Society of Chemistry, on the other hand, does actually do everything, and they do it all. Project Prospect, they mark it up, and they means that their journals, individual papers, are very highly searchable, and it must be costing them a fortune. But I think they are probably seeing the return on the fact that people access their journals. So it's beginning, and I think it will escalate, but it's really, the next few years will tell. So we have to get more papers out where we have done this.
And the Royal Society of Chemistry are very orientated around ORCID and the data science
Yes.
community, so they're really driving.
They bought ChemSpider and got it to be believable and workable. So there are now web services in chemistry that are available, which there weren't a few years ago. So it's beginning to happen. And it does mean, in Mark's work, when an identifier appears in the blog, it now can link to something, whereas before it can't. That helps people. The pictures appear. You don't have to redraw them. It's every time that happens, so it actually means that you save time. And that's the critical bit, because you don't redraw it. But tables are still actually a problem.
Please join me again in thanking Jeremy for a very nice presentation. Sir. And now I'd like to introduce our colleague, Michal Ondrejcek from NCSA, and continuing with the theme of tools for science. He's going to tell us about some important work that your lab has been doing related to software conversion and the registry you're making available. Mm-hmm. Thank you very much.
Thank you very much for having me. My name is Michal Ondrejcek, and I'm from NCSA. And I want to present this work of our group, the group of 5 people led by Peter Bajci, about the conversion software registry. So the overview, the work is sponsored by National Archives and Records Administration, which shows you like the importance and who is interested, in fact, in our work. So with the increasing number of file formats which are used, preservation for NARA is really important. And they are asking the questions like, will there be software to load the file in the future, or the specification of the future file format? You see, they want to know, they want to estimate. It's very important for them for preservation of the digital knowledge in future, to know at least what file formats will survive. Some of the file formats are proprietary, and that's the driving force of this work for NARA. So conversion, we just talked about, you convert file to an open standardized format to store, and you store it with the original. So what the format, how and which format it's going to be. The conversions, as you can imagine, we are talking about the 2D, 3D conversions. They result in some information loss. So that's not unusual. There is no universal converter. And of course, it's even worse that there are different domains, and they prefer the different type of formats. In 3D, for example, some formats are more tailored towards the preserving of faces or certain features. So how do we convert? We want to just convert many file formats. There are many closed proprietary formats, as I said, and there are large and complex specifications. The last point, for example, the step specification, the 3D format has hundreds of pages, and it's really time-consuming to write a loader. So this is the main reason why we started this work. Just we want to give some estimate of losses between, if you convert between formats. So we want to just estimate the information losses and give NARA some, it's not recommendation, but give NARA some estimate what formats are the best and what are the probable formats which will survive. So let me start with the 3D domain. Just this slide only shows that there are many software packages and many different formats based on the extensions, and so not only that, most applications, they support only a handful of imports and exports. They perform differently based on not only on the algorithm but on the purpose of the domain. So here I choose 2 different 3D packages, and you see the import. I don't know if you can know. You see the import and import and export format. So what we are doing with this, we just monitor, or we just write down these tables of import, save, open, and the formats. And we have the technology which can convert from one file format to another, which visualize this type of tables. User can choose the destination format and the initial format. And we give the user some kind of comparison tool, and we can measure the losses. So these are the technologies, and I will briefly go through the technologies because they are important for the database for the conversion registry we developed. So we have 3 layers. In fact, the first layer is called owGÞ, is the visualization based on the table of import, export, open, save. The second layer is the actual conversion done by the framework we call Polyglot, and the third is the comparison done by the program called Versus. So here is the input-output formats. You see the table, and we create some graph, and the lines are the edges between 2 formats, import-output formats. So it's visualized. So you can choose, user can choose the shortest conversion path. There is not always the direct path. So in this case, for example, you go from the red, you go from one file format, from step to Act3D, and you have to go through the 3 different software packages to get to the destination. So on the right side, you see the software packages, some of them proprietary, some of them open source, you have to use. So user can, for example, if the user has all of these software packages installed on the computer, with this type of visualization, he can just find the path, and with our polyglot server, user can actually do the conversion. So the second part is the conversion part, and it uses the third-party software reuse, the code reuse. So that's exactly what I said before. You have your software installed on your desktop computer with all licenses, and we only give you we give you the interface which will somehow drive that software. So most of the software packages are GUI packages, and there is not easy way how to operate them from some other program. So what we are using, we use the wrapper scripts. So there are different scripting technologies for different platforms, auto hotkeys for Windows, Apple script for Apple, or various shell scripts. And with the help of these scripts that our polyglot server calls the scripts, and the scripts actually operate on the software, opening and saving and catching up the exceptions. So that's the layer which we call software reuse layer. It exists as a service on the machine where the third-party software exists. Here again, I want to point out, it's your software, you have the license, it's on your computer, nobody can directly access it from outside. Client provides only the Java API interface, and many servers can exist of many machines of different platforms. So Polyglot it takes actually all of these different scripts, and it creates that one visualization tool, the graph for the shortest path. It searches the shortest path from one format to another, and it has no direct knowledge of the software which is installed on your computer. That one is done by the scripts. Now, I want to point out the scripts. We developed some nomenclature that we have the header, and all the input-output files are stored in the header of the script, and Polyglot looks into the header and creates those tables. The third layer is the comparison layer. You want to know, okay, I did the conversion, so how good the conversion was. So we have this framework for actually comparing digital object formats. They can be anything, and it depends on the measure, on the loaders. You program, you install, and then you can actually compare two files. We don't compare two files directly, because if we can do that, we would need loaders for all the formats, and then we don't need any conversion. Then we simply have all loaders. We don't need a third-party software. So what we are using is the A prime scenario, that we think statistically, if you do the conversion across many software packages, across many paths, you finally get something that is close to the losses for one particular conversion. So the tool weights all the losses, and then inserts those losses to the iograph, and iograph uses the losses as a number. It's a normalized number for the next computation of the shortest path. So this is actually what we have with this technology. Now, so far I've been talking about the tools we use. They are available, and you can use them on your own computer. There is no database behind that. So I will just go through this. This is like the scenario. You have the three software packages marked green. You have this nice iograph which can convert. It shows you from which file you can compare to which. Then you have the tool. You have the bunch of test files, and you run them. The green is the successful conversion. Red is the not successful conversion. Blue in this case is just in process. And then you assign the number, the losses. So you see from one format to another, we have 0.89, 13% loss. So all of these pieces of information we can store in the database, and then comes the idea of conversion software registry database. So what is it? It's a complementary database to registries such as UK Pronom or GDFR, which store the information about the file format. There is no similar service that we are aware of, and it's community contribution encouraged, so everybody can download Polyglot as a server and can run on his or her computer the conversion and populate the database. So the important part, the database focuses on the conversion, not on the file, not on the extensions, on the conversion software. So we want to find subset of softwares for specific conversion needs, and we want to find conversion between pairs of formats. So this is that's the design of the database. Again, 5 different parts. In the heart is the conversion tables. In order to characterize the files and the software, we have the software part with vendors and with all different information, like the version. We have the scripts which actually operate on the software, and we have the test files and formats, and we have the login and history part for editing the database. So this is how do you add the conversion to the database? So you just use the web entry, you specify the software and input, output formats based on the extension. Or you can download the script. You can upload the script directly. So we wrote a script manual, and we recommend users how to write these scripts, and especially the header of the script. So then the information is parsed from the header. So upon upload to the server, the table gets populated with all the input-output information. There are different types of scripts. The most important script is, of course, convert script. You can imagine many packages in 2D in images like EarthunView. It's an open source software, and you can use it in command line. So in fact, the script for converting for this particular software is pretty easy. It has a header, and then one line which says convert parameter 1 to parameter 2, in fact. So that's the full conversion. If that conversion doesn't work because it needs some third-party loader or something else, you can the script either deals with that, or it leaves it, and after some time the polyglot calls the kill script, and it terminates the software, and actually that conversion doesn't happen. So, or the measure is assigned to 0, there is 100% loss. So we have some other scripts are necessary for certain 3D software. We need monitor scripts as well, because those software packages are very complicated. This is edit. That's what user can edit all the information about software platforms, interfaces, formats, and so on. What about file formats? I said that it's based on the extensions, but extensions are not unique. You can imagine that, for example, pict in 2D, pict extension during the years has nowadays like 15 different formats have the same extension of TIFF. So we had to use some other identifiers. We still use extension when the extension is confusing. We actually decided to go with pronom PUID, which is a unique ID. We also record the Apple's ID, and we record MIME. So in future, we can use any of these identifiers. So as I said, PUID is different for different format versions. So here in this example, for example, the TIFF6, which is the newest or the current description of TIFF, has one PUID, and it's distinguished from for example, GeoTIFF, which has the same extension. We have test files, bunch of test files, and we are validating the files using the Unix command and Georgia Tech Research Institute file type identifier, which extends the Unix file format. You can search for software. As you can imagine, you type the input format, you type the output format, and it gives you the direct path, or you can go through one hop, what we call one hop. So you use some intermediary software. So in this case, you see how many software is there. The shortest path from A to B. So we use the algorithm mentioned here. It's the lowest cost, the shortest path between one vertex, with edges defined by some measure. And now the measure can be anything, actually. We also use subjective measure. When you edit software, you can rank it. The main reason for this was actually that the users inserted a lot of demo versions, so we assigned like lower ranking to demo. It wasn't like, at this moment, we are not capable of ranking software like to bad and good ones. So, so we have the subjective measure and we have the quantitative measures within the domains. So in image domain, we implemented two only. We implemented normalized cross-correlation measure and histogram distance measure. In 3D, we have more. And we are thinking about document comparison and audio comparison in future. Everything actually driven by the sponsor, by National Archives. So, and then user can specify, this one is not implemented, but in the near future, user will be able to specify measures which they like, or we will provide some kind of linear combination of these measures, and you can switch between measures. So what is important for you and for your domain. And these measures, they get stored in that graph, and the graph will show you the shortest path. So when you search for the now I'm showing the IO graph within the database. So it's a Java applet, and now it doesn't run on the script header, it runs on the actual input in the database. So it's taking all the information in the database. So you see you can again go through different hops. There is no path here. Or you can choose domain from. So there are all of these files which are available for that particular format to be converted to. Or you can choose, or you can combine it. You choose more formats, and you just get all the formats, which in this case these red formats can be converted to again. So future direction is compiling known good data for different formats, systematically measuring information loss across software and formats. It's a very time data intensive task, so we think that the community approach would be the best. Ranking software based on performance and integration of CSR and polyglot. So this is important. The database now is populated. It shows the shortest path, but it's still not integrated with that conversion software. So in other words, the conversion framework still takes the information from the scripts and from the software which is installed on your computer. It doesn't go to the database, that conversion part. Summary. Currently, we have 2,000 software packages, 1682 format extensions, and 233 conversions. The giant number of conversions, it's actually based on what I would call theoretical import-export. It doesn't necessarily mean that all of these conversions are or can be performed. So this one is up to, some of them will be red in that graph I showed you. So there is no similar service. It's free. We believe in community contributions encouraged. So I want to mention that 2,000 software packages, we didn't put them ourselves. We work with NARA, with National Archives, the research branch of NARA, and they hired students over like summer interns. So they populated the database, and we were like leading that effort. Thank you very much.
Thank you, Michael. Are there questions for Michael? I've got one. What has the usage been? Obviously, it's targeted towards NARA, but has there, you've got these numbers here, but in terms of number of users or types of institutions?
Types of institutions, NARA was the major part, but we had not a couple of, we had, I think, five. I think 5 users from other 5 companies interested in this. And how I know, I see the login. So those were people logged from different companies they were using. We had some feedback from them. One of them was actually one of them was really difficult because the guy said, "Are you going to store formats which don't have extension?" So how? So what? We can't do that. So he mentioned that there used to be some kind of really old formats which don't have extensions, for example. So yeah. So I would say 5 users, 5 different users.
And that was my next question was, how many of those were like memory or cultural institutions like NARA versus corporations? Or were they
I don't.
Were there software companies, or were there companies who have backlogs of certain files?
There was IBM, for example, but I don't know what department it is. So I think it's like NARA. NARA itself is the research part. So what IBM, who in IBM, I don't know that. So.
When looking at the results that you've then got, are there any really surprising things where the direct conversion would be quite lossy, but if you go through, like 10 hops or something, that you actually get quite a good conversion? Have you seen anything like that?
No, we haven't seen that. And we are actually, to be honest, we are not even there. We don't have that many losses over 10 conversions. So that part is, we ran the tool, but we don't have the statistical results. So the numbers I have are, I think, this is the statistics of low numbers. So we have to do really big data set, choose the images, what images, for example, in image, like what images. Are we going to create our own synthetic images? Or are we going to use big collection? Kodak has nice collection of colored images. So what approach is better? So that will be my job the rest of the year. Yeah.
Well, this has been a problem for a really long time, so I'm glad that somebody's doing something about it. My question, actually, I'm on this National Academy Board on Research Data and Information, and the board is very interested in things like this being available in the long term. And so between NARA and I think you had some other government agencies sponsoring this, is there some kind of written guarantee that this project is going to exist for a long time and is sort of the official...
He'd like to talk to you about that.
Yeah. Yeah. No, but it's
Wow.
very serious, right? You said you have no competition, right? You said no one else is
No, we
doing
are
this,
not aware of. But
right? And so this is a unique service that...
first of all, NCSA works on the software developed in NCSA is open source. That's the policy. So this part will be there. I think NARA wants to make it available, and the Pronom and the GDFR are actually open source as well. So probably your question is the support mainly then,
Right.
like, are we going to support it? We don't know yet. And there is a discussion now, who's going to host it? Is it going to be in NCSA, or NARA will host it, or will it be like open and everybody will be able to download a database? And then how are you going to...
And what I'm particularly curious about, I'm sorry, but this is a very strange question, but about a dozen years ago when I got my very first digital camera for personal use, okay, I became very worried about what was going to happen to people's personal photographs. And I realized that there was a business model for some company like Kodak or something to promise you eternal migration of your formats. And so if companies decide, even for things like that, for consumer use, that they're going to somehow use all of the stuff there, is there a licensing problem where it's open source and they can
This
just
one is
use
open
this?
source. We get
Right.
question about, can you do that? Can you actually reuse the third-party software? That's the most common question. I mean, the database and the technology behind that will be open source for sure.
But if they want to set up a service where they own a copy of the software and they do this for you, then
I think it's legal. I thought about it a lot, and actually I read more licenses than I read before, and I don't see anything which actually would like prevent that. It's on your computer. We are not accessing it directly. If it's locked, it's your software. You have the license. It's like that example, like your VHS, for example. You want to transfer it to digital. You go somewhere and you pay guy $20, and what is he doing or she on her computer? Is it legal or not? You just buy it for $20. You get your digital format back. So it's a pretty common business model. So this is the same, except you don't have to pay. So, I mean, if you send us email, I will be able to answer those questions more. I mean, the main problem for us now is exactly: is NARA going to host it? Are we going to share the database itself? Probably we will, but I don't know that yet. And what's going to happen when you have different instances of the same database? How are you going to merge it? So there will be one center which will be hosting that. So that's in negotiation now. But it will be publicly available. We support 20 years from now.
so
I don't know.
We need to go with the next session, but if there's one last question, maybe from John.
Yeah, it's John Kunze from the California Digital Library. I'm assuming that the complementarity with GDFR and Pronom is going to encompass at least some sort of ability to link to the canonical names for the formats. And I just wanted to make sure you're aware of the Unified Digital Format Registry, the UDFR project, which is going to bring together Pronom and GDFRs.
Yes.
So it'd be really great if that there was a continuity of complementarity, that these could actually link to each other.
Mm-hmm.
So this
But
project's
UDFR
about to start up, and we'll be hosting it. So maybe I could put you in touch with.
I think there are this. I'm pretty sure you've been in touch with somebody in NCSA, because I think NCSA was behind some of these, or University of Illinois, maybe not NCSA. So, but my understanding is UDFR is the extension of GDFR, isn't it? It's just the.
It's essentially it's unified. So Pronom and GDFR
yeah.
together. Yeah.
I personally hasn't been involved with any, but I'm pretty sure that Peter Bajci is the contact person, and he.
Okay.
Sorry, thank you very much, Michael.
Thank you.
Michael, can I help you out with homework?